In this lecture, we're going to be going through the types of specimens that you're going to be getting and associate them with the types of infection, so that you know that depending on where the infection is located, you're going to get a different type of specimen and how we would treat that. One of the biggest things that you have to get used to in microbiology, and it takes time as you're working there, is you have to learn what to expect at a particular site, what the normal flora is. So these are the organisms, and you can see by the cute little picture here, 99% of the things that grow in you are perfectly fine, but a few of them are nasty, angry little creatures, and they can create infections. Even those 99% that normally live on your skin, they can produce opportunistic infections, as you can see here, which is they occur mostly in immunocompromised patients. And it basically is somebody here, the 99% that don't cause you any problems, they gain access to a site, a new and different site where they thrive. And so the good thing about them is that they're usually a lot easier to treat because they're kind of weak, and they're really not as angry, I should say. But they normally they do live as part of your body. The other thing that happens when you have patients in beds who, they're immobile, their normal hygienist is affected. When you put people in a bed or when you attach outside objects to them, catheters, tips, lines, things like that, those things become a great site for something called biofilm. Biofilm is a group of bacteria that live together symbiotically in a community, and they're not living on you, they're not attached to you, but they live over your normal flora and over potentially a wound site. So we need to be able to discriminate between normal flora and biofilm, and all of those and a pathogen. So biofilm, usually as a contaminant, will look like many mixed bacterial flora. You'll see lots, whereas normal flora, everything's pretty evenly distributed, and you don't get a ton of it on your plate. So the normal flora becomes what we would call the usual suspects. These are the guys, they're not the usual suspects. The usual suspects are those organisms that we know are known to cause an infection at a particular site. Okay, so for example, most wound cultures are caused by Staph aureus. Most urine cultures are caused by E. coli. But most is a very vague word. That could mean 51%. So you still have lots more that you have to look at. But when you look at wound cultures, you're going to look at a lot of Staph aureus. When you look at urine cultures, you're going to look at a lot of E. coli. Okay, just that particular organism does particularly well at that kind of site. Because we know what the usual suspects are for a particular site, we can choose the plates in order to ensure the growth of that particular organism. So you would make sure that you have a gram-negative plate to catch E. coli in a urine. You make sure you have a gram-positive in order to catch a Staph aureus in a wound. Okay? And then we have to recreate the conditions atmospherically, nutritionally, that those organisms need. Okay? Staph aureus can grow at ambient air, but it prefers some CO2. So we put that in the CO2 incubator. The enteric plates, the ones that grow gram-negatives and tell you lactose fermenting or not, those ones you can't put in CO2. The CO2 is going to change the pH, and the pH is vital for our reading that. So you can't put those in CO2, it ruins them. Okay? So what we're doing, remember in the microlab, we know that there's something bad growing in our patient. We actually want to help it grow in the microlab so that then we can learn to kill it. Okay, those are the things. It's a little devious, but that's what we're supposed to do. So we want to make sure that we choose the right media for what we expect to grow there, that we have that media in the right temperature and atmosphere, that the pH is maintained, and all of those things are in our control. So we make sure that we take care of them. Now, the usual suspects, what do we find at various sites? So this is, you can see, a very comprehensive list. You've got, depending on the source, is it upper respiratory? Is it lower respiratory? Is it the skin? Is it the GI tract? All of these places are, they have different normal flora, and they have different, what we would call the usual suspects, the things that we would first think of. But it's from these lists that we determine what plates we're going to use. So we know, if we just look at one group, okay, let's look over here at the upper respiratory tract. We know that the usual suspects include Staph aureus, that can grow there, strep, in particular some of the beta streps and strep pneumo, the alpha streps, those grow there. We know that Haemophilus can grow there. We do know that anaerobes live there, but we don't worry about them causing an infection there. So based on that, we want to choose plates that will grow these three things, definitely grow these three things. So I know that Staph and strep can both grow on a blood plate, but I also know that Haemophilus can't. Haemophilus is a fastidious organism, and it requires a chocolate plate. So whenever I set up an upper respiratory, maybe a nasal drainage or something like that, I'm going to make sure that I include a blood plate and a chocolate plate, as long as I'm looking for those things. And that's how you kind of build your media. Each hospital will have that already built. You don't decide. The hospital has a procedure built in, and they say these are the media that you choose. Can you add to that if you see something that makes you change your suspicions? You absolutely can, but you cannot take away from it. Okay? You can always add an extra plate, but you cannot take away from the plates that the procedure calls for. What are the transmission routes for all of these different types of organisms? Well, some of them, through just living your life, influenza virus is an aerosol that you breathe it in, and it sets up shop, hopefully just in your upper respiratory. If it sets up shop in your lower respiratory, you're not feeling too well. Okay? Oh, I'm getting tea everywhere. Oh my goodness. But some of the other ones maybe you're not as familiar with, okay? Most of them you probably are. Herpes, that's a direct contact mechanism. And where does it come from? Well, from another human, okay? Now, the influenza virus here, we say that the source is an infected human, and that's because that's usually the case in the United States. But actually, it does come from animals. And in farming country, you do have to worry about your animals, and you can get it from your animals, like birds and stuff. In birds, interestingly enough, it's a gastrointestinal issue. So it's really, you come in contact with bird poop, and you can get influenza. Syphilis and gonorrhea, you could see the different mechanisms. I don't have to read all this to you, but you should be aware of, based on the portal, and so where's the infection that we're going to have. So these are mostly this line is, this column, is it a respiratory, is it a skin? Well, how do they get there? Those are the things you should know. So we're going to go through each of the different culture types and talk about what to expect from them, okay? So blood cultures is one of the ones that we receive a lot of them in the lab, mostly because we do 3 on everybody. But we do receive a lot of them, and you should understand what to expect there. It's your blood. Your blood is a sterile site. So nothing should grow there. That's the good news. Most of the blood cultures that we receive, we will report normally as no growth after 5 days. Yay! Nothing grows there. The patient does not have septicemia. Okay, so that's a good thing. The problem is, if something does gain access to the blood, it then has access to every single organ in the body. So you can end up seeding other organs with these bacteria if the blood becomes infected. So that's very important. That can lead to shock, organ failure, DIC, which stands for disseminated intravascular coagulation, where basically your coagulation system goes a mess. You start throwing clots, but also bleeding all over the place. And that, of course, could lead to death, obviously. When we're doing a blood culture, we're looking for two different things. We have two different terms that you have to be aware of. Bacteremia, which technically means bacteria in the blood. Now that can be transient. We've talked about dental work, things like that. After you brush your teeth, if we test you, there's a transient time where you might have bacteria in your blood, but your white cells take care of that. Okay, so hopefully most bacteremia is transient for you. Septicemia is a little bit different. Not only do we know that you have bacteria in the blood, but the bacteria actually reproducing, they're multiplying and reproducing in the blood, and as they do that, they'll be producing toxins, which obviously are going to cause significant harm to the host. Most septicemias result from some kind of IV catheter, some other portal of entry. It could be the GI tract. That's unfortunately, if a patient develops what we call leaky GI, or they have holes or bleeding in their GI tract, that can lead to the bacteria from your GI tract gaining access to the bloodstream. Other ways, if you get pneumonia, because the lung has such an intimate contact with the bloodstream, bacteria can pass over from pneumonia and it can turn into septicemia. Any abscess can become septicemia, as well as a surgical wound, obviously. Those are deep wounds. Obviously, septicemia is a possibility for those. So those are your risk factors for a positive blood culture. They actually end up being some of the easiest cultures to work on in the lab, because if it grows, you work it up. They're just some of the most important, because these ones, if anything grows there, obviously the patient's at risk of death, so it would be a critical lab result. So you got to call the physician and notify them. When we collect blood cultures, we use aseptic technique. And it's important that you understand the difference between sterile technique and aseptic technique. In sterile technique, you want to eliminate all bacteria from the system, which is a good thing in manufacturing, stuff like that. However, in microbiology, if you do that, then we won't be able to grow up the organism we're looking for, so that then we can learn to kill it. So aseptic technique is actually what we use in microbiology. We don't want to add additional bacteria to it. We want to maintain the populations that are at the site. And so we use aseptic technique in order to collect specimen, any specimen really, but in blood cultures we have a very particular procedure. And you can see, I have the procedure written down here. Some of the things are a little bit subtle, but you see the big swab that the person has here. That swab, you start in the center and you go in concentric rings outward from there. And then as you do that, what you're doing is you're killing from the center outwards. You want to use a very large area. You can see how large this area is for this particular procedure. The order of the things that you're going to use: 70% alcohol. You must let it dry. The alcohol on its own doesn't do anything. It's the process of that alcohol evaporating that actually causes the death of any organisms there, okay? Because it takes the water with it and it dehydrates the bugs and kills them. So after you allow it to dry, do not wipe it off with a gauze. Allow it to dry. Then add the povidone iodine, which is the ugly stuff. Also let that dry. It might take a little time. Let it dry. Okay, that's how it works. It actually has to penetrate the bug, the bacteria. Then after that, again, let it dry. Let another round of alcohol. Let that dry. This takes time, and people don't usually want to take the time. Okay, but this is how you prep the site. One of the other things that you have to remember is that the tops of these bottles are not sterile. They may not even be clean. They've been sitting in some closet somewhere. So when you take the caps off of these bottles, take this alcohol swab and put the alcohol swab to the top of that and let it dry. Okay, so your phlebotomy, you're going to make sure that you've used cleaned bottles. You're not worried about the base, just the top of the bottle. Okay, but make sure that you actually wait for these things to dry. We do receive 3 sets of blood culture bottles. There is some controversy on this, and there's some studies that have said that maybe we really don't need 3 sets, maybe 2 sets is enough. Hospitals are kind of going back and forth on this. The key thing is, though, if you only receive 1 set, you're not doing anything. You're not helping the patient. We must receive, at this point, we're still saying 3, but we must receive at least 2. And that's because you do have the risk of contamination. And so we need to be able to compare the multiple cultures with each other. Okay. Now, the blood culture bottles that we receive is an anaerobic and an aerobic one. Okay. There's also a pediatric one for short draws, but basically we have an anaerobic and an aerobic that you'll receive on almost everybody. The aerobic is a brain heart infusion broth. And TSB. It depends on the manufacturer, but it's just basically a nutrient broth. Then the anaerobic broth, it also has a TSB. It has a nutrient broth, but it has reduced oxygen, so it has an indicator in it, or something that can reduce the redox potential so that you can reduce oxygen in that particular bottle. Okay. Now, anaerobic blood infections are much more rare, and actually they're on the decline as we become better at treating bleeding ulcers and things like that. So if it's impossible to get both, fill the aerobic bottle. That's the more important of the 2. Now, there's a media called SPS, and this is it over here. It's a yellow top tube. The SPS, or sodium polyenethyl sulfate. This is for drawing blood when you don't have the blood culture bottles. You want it for a blood culture, but you don't have the bottles. So you sometimes get these from doctor's offices because they don't usually maintain the bottles all the time. So you might get this from a clinic, an outer clinic, or a doctor's office. They might send this to you. Basically, the SPS obviously has an anticoagulant in it, but it'll also have chemicals in it that will help to counteract things like white blood cells, complement, and also antibiotics. So the other time you might receive an SPS, now you can receive an SPS if a patient has already started antibiotics, but they come up and they think they're still septic. There's also blood culture bottles that have charcoal in it, which would remove the antibiotic, neutralize the antibiotic, because obviously we want to grow it, so we don't want the antibiotics in there. The volume that you're supposed to draw on any blood culture bottle it's a 1:10 ratio. Basically, you need to draw in these bottles here. You need to draw between 8 and 10 milliliters of blood. Okay, there is a measuring. There's a little gradation on the side of the bottle so that you can watch the blood go in. If you can't get that much, then the idea would be to use pediatric bottles because they're designed for those short draws. If you can only get 3 or 4 mils, okay. And then I just mentioned before there are additives. In some bottles we have resin or charcoal. Those things do a couple of things. The charcoal is really there just to remove the antibiotics, but if it has resin, that'll also remove the antibiotics or neutralize really the antibiotics. But it also can provide a solid surface if the bacteria does better growing on something. So that's a possibility. So what are the things that we expect in the different sources besides blood? Well, let's go to the upper respiratory infections. This would include your sinuses, your throat, your nasal cavity, epiglottis, and larynx. One thing that's important to state here, these are all just if that's just the source, if the source just says sinus or if it just says epiglottis or something like that. If it says abscess at this site, that's totally different. You treat it like an abscess. So you have to make sure you know exactly what you're collecting here. Because you can get abscesses at these sites, and you have to screen for anaerobes. We do not screen for anaerobes in upper respiratory, normal upper respiratory tracts sources. Now here's the thing about the upper respiratory. Most of the upper respiratory infections are caused by viruses. Okay, rhinovirus, coronavirus, arenavirus, those things. So most cultures, it's going to be negative. Okay, patient wanted a culture, doctor does the culture. It's negative, it's a virus. You get over it in about 7 to 10 days. That's kind of the way it is. Okay. However, there are some bacterial usual suspects, as we said, and by far the most common bacterial cause of infections in the upper respiratory is Strep pyogenes, which is a group A beta strep. So when if in your hospital, it may be that you have a procedure that says that for any throat specimen, maybe, or upper respiratory, you're only going to screen for Strep A, since that's the most common one. And I've worked in hospitals that they only screen for Strep A. And that's a legitimate thing, as long as the physician knows that that's what you're going to do. Okay? And Strep A is something we do have to screen for, because actually some of the side effects of a Strep A infection are pretty significant. They can cause rheumatic fever. It can cause glomerulonephritis. These are rare, but in a patient who has had rheumatic fever in the past, these are autoimmune. Well, they're not really autoimmune because it's not a self, but these are immune mistakes that occur when you have an infection with Strep pyogenes, where the antigen that your antibodies are reacting against happen to share some similarities with antigens that are present in the case of rheumatic fever in the heart, in the case of glomerulonephritis in the kidney. And so we can treat you and get over that infection, and the antibodies will wane, and the damage to your heart and your kidneys will cease. Not necessarily reverse, but they'll cease. However, you get another strep infection, the same problem is going to happen. So we must screen patients who've had these things before very closely to make sure that they don't get them again. How do we collect them? Usually with a culturette. And these are the swabs. You've seen them before if you've ever had a throat infection, right? This is what the nurse comes at you with, okay? And just to remind you of the structure here, nose, pharynx, larynx, trachea, bronchi, and then down to the lungs. That's the order, okay? So don't forget your A and P that you took, okay? Now, there is one bug that is increasing. It's on the rise, and mostly because people don't get their vaccines, okay? So if I decide not to get my vaccine, certainly I'm at risk for getting Bordetella pertussis, but I also can bring it around with me to other people who may have gotten their vaccine, but their immunity may have waned. Maybe they're going to Sloan Kettering and they have cancer. Maybe they're elderly. Maybe they're very young. These are all the cases. And so it does increase in the population, not just of the unvaccinated, but also the vaccinated. And so Bordetella pertussis is a special case. The doctor has to know that that's what he suspects. It's a very distinctive clinical symptom with the whooping cough. So that actually requires specialized media, and you have to really make sure that you know that that's what the doctor is looking for, so that it can be plated to that media and sent for culture. Most labs don't do pertussis culture in-house. They send it out for a reference lab, because once it grows up, they'll just do the nucleic acid testing to confirm its ID. So the things that we receive, you've seen this before. You've had this done before. You see the tongue depressor, hold down the tongue. We don't want that tongue to get in the way. And the swab has to go all the way back. See these swollen parts here? Has to get in there to get the swab right against those nastinesses, okay? Now, I doubt that you can open your mouth this wide. This is a great cartoon. So it can be difficult to see and to get in there. If your patient doesn't gag, you didn't do it right, okay? So that's the important thing. This is the key one that you use for the most part, and you'll get it. You can see it's a double swab. We can use one swab for a rapid antigen test, which is a serological test, and we can use the other swab to back up any negatives with culture. If the serological test is positive, we don't have to go any further. We know the patient has Strep A, and the doctor can treat it. However, the serological tests are not all that great. Well, they're good, but the Strep A antigens, they're much too varied, and they won't catch every Strep A. So we do have to back up every negative with a culture, which is something many pediatricians' offices don't know. So if you take your kid and they do a rapid strep, make sure if it's negative that they back it up with a culture. For the nasopharynx, though, here's hopefully you haven't had to have this done, but this is a different kind of swab. You can see the tip is much thinner, and this is actually made of wire. It's very flexible, and you need that for a nasopharynx because you're going up the patient's nose, you can see here, and back into the pharynx. Okay, so it is uncomfortable for the patient, but it's something that we do. We do it for obviously we do it for pertussis cultures. We also do it for influenza, because influenza normally lives in the upper respiratory tract. So we get swabs down for either antigen testing or sometimes culture, mostly antigen testing for flu, using a nasopharyngeal swab. The lower respiratory tract, a little bit different. Once you go below the larynx and through the trachea and the bronchi, those bronchioles, and into the alveoli, there's not supposed to be anything living there. Those are supposed to be clean, okay? Now we get stuff because obviously we're breathing in all kinds of pollutants and bugs and everything, right? So we do get stuff which can cause several diseases. You've all heard of COPD. They advertise it all the time. You can get acute or chronic bronchitis where the infection is in the bronchi, which is this part here, okay? And it's supposed to be sterile, so anything that gets in there could potentially cause an infection. Acute bronchitis, again, it's still usually viral. An example would be respiratory syncytial virus, or RSV. Also flu. Some strains of the flu are able to survive at the lower temperatures, at the higher temperatures lower down in your respiratory tract. And if you get one of those, you have an increased risk of getting pneumonia, okay? Chronic bronchitis, where there's an inflammatory response, it's kind of constant, that's usually not bacterial either. That's usually caused because smokers are breathing in stuff that your lungs don't like, and so the bronchi are inflamed a little bit persistently, okay? The most serious lower respiratory tract infection is pneumonia. And the most common bacteria that causes that, strep pneumo, okay? These, if you're sick, if you have acute bronchitis, chronic bronchitis, pneumonia, we can tell that you have those. A pretty good sign is the fact that you're going to start producing something called sputum. Sputum is only produced by adults, not produced by infants, right? So you can't get a sputum on an infant. And it's only produced when you're sick. Normally, you don't produce sputum at all. So getting a sputum specimen is difficult, but we have to depend upon the patient to get it for us, okay? Basically, we want to make sure, since it is so hard to get a sputum, that the sputum specimen that we get is a good specimen. And so we're going to look at it in a gram stain before we culture it. We don't culture the specimen until we look at it in a gram stain. And we look, this is an example of a sputum culture, a sputum gram stain here. And what you're looking at, you see these, this cell here, large cytoplasm, small nucleus, right? That's an epithelial cell, okay? Epithelial cells would not be, these squamous epithelial cells would not be found deep in the bronchi and the alveoli. You'd have columnar ones down there, right? So if you have a lot of those epithelial cells, squamous epithelial cells in your gram stain on a low power field, more than 25, then it's contaminated with saliva, and we do not culture that, okay? The point is that we want to force the doctors to use a good specimen and not use bad specimens to diagnose pneumonia, okay? They really should be using imaging. They should be looking for changes in the lung x-rays. But they do sometimes use the sputums, and we want to make sure that we're not giving them bad results. If your specimen does have more than 25 squamous epithelial cells per low power field, you cancel it. It's actually required that we cancel it. We are not supposed to culture that, okay? Even if we see lots of interesting things, it's still spit. Get us a better sputum. And we would prefer first morning sputum if you can get it, okay? And remember, if the patient has pneumonia, they should be producing a lot of it. So here's the actual cartoon or whatever that we use to show patients how to collect the sputum. Basically, to get that sputum out, they should have a pretty deep and pretty painful cough. We want them to try and expel that sputum directly into the cup. So we want them to cough open-mouthed into the cup, okay, which is, it goes against everything you've learned, right? You learn to cover your mouth, to, you usually close, you don't want things coming out, but in this case, we want you to cough open-mouthed into the cup. It's a sterile cup, and then the nurse will bring it to us, and we can collect it. You can understand what probably happens most of the time is that the patient coughs into their mouth and then spits it into the cup, and that's the problem. We cannot salvage that specimen in that particular case. So what about patients who can't produce a sputum? Maybe an infant, or there might be something else, or they can't give us a good sputum. Well, in those cases, we do what's called a bronchoalveolar lavage, or a BAL. The BAL should be sterile. The sputum, we don't expect it to be sterile because it's passing through the trachea, the larynx. It's passing through all those things, picking up bugs. But the BAL should, and that's because it's collected by the physician. And you can see here, it's pretty awful. The physician inserts a tube down the patient's throat and then washes out the sputum and the gunk that they find down there, collecting it in this wash trap. Okay, that's what it's called, a trap, or that's what we call it in the lab. Okay, this should be sterile, and it's not unusual for a lab to treat it as if it was sterile. Okay, so this one you don't expect to find lots of contamination in, and so that's important for you to differentiate between the type of specimen and what's the normal flora there. Normal flora in a sputum, that'll be normal respiratory flora. Some of the upper respiratory tract stuff will get in there, but in a bronchial, in a BAL, that shouldn't be there. Now we're going to move on to the urine. So the UTIs, or the urinary tract cultures, remember, UTI, urinary tract infection, can be anywhere from kidneys, bladder, ureters, and urethra. Okay, so we have to we collect the urine that help, and there are markers in the urine that help us determine if where that infection is. Okay. Now, a UTI, normal UTI, is usually cystitis, which is a bladder infection. Okay, that's our most common UTI. Usually some bacteria from the colon gains access to the urethra and moves up into the bladder. Okay, usually requires some kind of illness on the part of the patient, primary illness on the part of the patient, or a loss of immune status. If the infection is able to move up the ureters into the kidney, now we're talking about a different thing. That's called pyelonephritis. That's an infection of the kidney, of some portion of the kidney. Okay, we can still test for pyelonephritis using a urine. We combine both the urine culture and the UA results, because there will be signs in the urinalysis that this is actually pyelonephritis and not cystitis. And those are the things that we look for. We have to combine the two. A urine culture without a UA is not as helpful. Okay, urine is obviously very easily collected, but we do get both sterile and non-sterile specimens. Our most common patients would be, they're more common in the elderly, more common in women. So we have to make sure that we educate our patients as to how to clean the site and collect the specimen properly. Okay, our most common pathogen is E. coli. Our most common gram-positive is Enterococcus. Both of those are gut flora, and they just gain access to a place where they're not supposed to be. Okay. Unfortunately, in hospitals, UTIs can be easily acquired. So many UTIs in the hospital would be nosocomial in nature, and that's usually because of the catheter, the stupid catheter that they put in. So we try not to use catheters, try and keep patients mobile, keep them using the restrooms, or at least a bedpan. but unfortunately those are less comfortable for the patient sometimes if they have pain moving, and the bedpan's never comfortable. So catheters might be used a little bit more often than they probably have to be, and then a nosocomial infection is a high risk, okay? Now, even though urine is created sterilely inside the body, it's not released sterilely, okay? We don't collect it sterilely. So we remember that we don't consider urine sterile unless it was collected directly from the bladder by a surgeon, or if it's a nephrostomy urine, because that came directly from the kidney. So those didn't pass through the urethra. So in that case, we would consider it a sterile specimen, okay? And you can receive that in a cup. Here's a cup. Many hospitals are asking that they transfer this into the specialized tube so that it has, we get extra time to set it up. And when we do set it up, we always use a quantitative culture method to set that bad boy up, okay? Now, it is easily collected, but it is as easily as it is collected, it's very difficult for us to get a good specimen. And again, that's because we're not collecting it. In fact, even the nurse isn't collecting it. The patient is collecting it, and they're less educated about how to do things, and they're sick and they're not feeling well, and it's hard to get somebody to listen, this is what you need to do. And then, okay, they listened, but do they understand? So these are the things that it's really up to us in the lab to educate the nurses, so the nurses and the aides can educate the patient, okay? And the key thing really is proper cleaning, in particular for women, because there is so much normal flora in the site, and so it does have to be cleaned properly, but difficult to do, okay? Our ideal specimen is a first morning midstream specimen. That's what we really want, okay? So again, what's a midstream? Midstream is when you the flow of urine starts. You don't take that first bit of urine, you allow that to pass, and then you collect urine with it. It's a messier way to do it, but that's what we ideally want, okay? There are other specimens, suprapubic tap, that would be a needle inserted over the pubis bone so that you go directly into the bladder. That obviously would be considered sterile, okay? And then we have our catheterized specimens, which include a straight cath or an indwelling cath. A straight cath is when the catheter was actually inserted to obtain the specimen, and then it was immediately removed. So this catheter doesn't stay in there, as opposed to an indwelling catheter, which it's a Foley cath. It stays in there to collect this, urine over time. The straight cath is fine. We collect those, you can get those all the time, and you might have a increase in cells in that on a UA, but you shouldn't have a ton of contamination fluid. The indwelling cath, however, does become colonized by biofilm very quickly, and so actually within 24 hours. So if the indwelling cath has been in place more than 24 hours, you have the risk of contamination, and your hospital has to include in the procedure how long the catheter can be in place before you have to change it for a urine collection. Okay, hospitals all differ. Most hospitals use 5 days, even though we know that there's going to be biofilm after a day. Okay, but they just don't want to bug the nurses that much. And then finally, we have the pedia bag, which you have seen before. It's absolutely the worst. Okay, we get terrible specimens from the pedia bag, but it's the only way to collect from an infant. They just don't, they don't do what you want when you tell them to. But you get tons of contamination in a pedia bag. So because of, you're going to get fecal flora in a pedia bag. So because of that, you have to make sure that you're not over-reading a pedia bag culture. The quantitative culture setup that we do, basically we're trying to give the doctor a colony count, okay? So the number of colonies per milliliter of urine is what we want to give. So that means we have to make sure that we set it up with a calibrated loop. We can set up if we set up the urine with a 1 microliter loop, then we're going to multiply the number of colonies that we see by 100. I'm sorry, by 1,000. It's a 1 microliter, so it's 1/1000th of a milliliter. We multiply by 1,000. So if you see 5 colonies, it's 5,000 per milliliter. 50, it's 50,000 per milliliter. Okay, and for the most part, we round up for the most part. Okay, to 5s or 10s. Okay, of thousands. In some cases, in particular in sterile urines like a bladder urine, because a 1 microliter loop will actually cause, you can have sampling error in that. It's usually not a major issue in urines because if you have a UTI, we're going to find it with 1 microliter. But in a bladder urine, they want to make sure that they take a larger sample. So they use a 10 microliter loop. And you just have to remember to change your factor that you multiply it by. If you use a 10 microliter loop, then you don't multiply by 1,000, you multiply by 100. Okay, very simple. And if you see confluent growth, if you look at this plate here, you're probably saying, how the heck am I going to count all those colonies? But if you see here, there's not really separate colonies here. Look at all this confluent growth. When you have confluent, or when the colonies merge together, when you have confluent growth, your colony count automatically becomes greater than 100,000 colonies per mil. Okay, you definitely have a UTI in this particular, if it's pure. That it's a sign, that's diagnostic. Okay, but we have to make sure that we watch out for contamination. So if your McConkey plate has what you can see are 3 different colonies on it, that's not from the urine. 3 different gram negatives at greater than 100,000 each, that's just a mess. Okay, that most likely got contaminated by fecal flora, and they have to get another urine. Okay, so, and then the procedure in your hospital will determine, once you know, okay, in this case here, we have greater than 100,000 gram negatives, and that's a great preliminary report to the physician. But what does my hospital say that I need to do? Well, if it's pure, then my hospital will say, you have to work that up, means an ID and a sensitivity. But your hospital will write the procedure that will determine what number of colonies is your cutoff. Do you only work up when there's 100,000? Or do you work up when there's 50,000? What do you work up? Okay, it depends on the hospital. Each hospital will have their own procedure. Most doctors don't think it's a UTI unless it reaches 100,000, but clinically, from our perspective, if it's over 50,000, you probably do have a problem. Now the stools. The stools, again, a little bit different. We're not really trying to grow everything in a stool because, my goodness, that would be hard to look for. We're just trying to grow the pathogens in a stool. And it's a pretty limited list, okay? So here's the list. Look, it's pretty short. Campylobacter, Salmonella, Shigella, Vibrio, Aeromonas, E. coli 0157. There's a couple of others as well. Staph aureus and yeast are 2 others that we're going to try and grow. But these are the main guys. These are the usual suspects, okay? So these ones, we have to collect the specimen and put it in a, here's your culture and sensitivity container. You can see it says CNS here, culture and sensitivity, whereas this part of it is for, this part here is for O&P. So the specimen will come in it should come in a Kerry-Blair. That CNS container has Kerry-Blair media in it. And what that will do is it will inhibit the growth of most bacteria, but it'll allow these guys, these troublemakers, the room to grow. Basically, what it does is it ensures that these guys are not overgrown by everything else, okay? Now, all of your outpatient stools should be coming to you in this Kerry-Blair medium, but sometimes you receive it just in a sterile cup if it's from an inpatient, which is fine as long as you received it very quickly. If it was sitting up on the floor for whatever reason for 5 hours and then you receive it, That's really not an acceptable specimen. The normal flora would have overgrown anything that was there. So either collect it in a sterile cup and send it to the lab immediately, or put it in Carrie Blair, and it increases our time. We have up to 48 hours to culture that. If you refrigerate it, we have even longer. Okay. Now the stool cultures itself, again, we're kind of changing our perspective. We're not trying to look at each colony and saying, I know what that is, I know what that is. What we're doing here instead is we're trying to say, oh, that's not salmonella, I don't have to worry about it. It's not Shigella, I don't have to worry about it. And so that's why we use a whole bunch of different plates. All of them have the capability of growing that small list that was there, and that we can recognize them on those different plates. What we have to weed through is all the normal flora. So look at all this normal flora. You've got lactobacillus way up in the small intestine, and then you add strep the way up in the duodenum. Then you add strep lower in the small intestine, and then look at what happens in the large intestine. My goodness. Now many of these, you can see the Bacteroides, Peptococcus, Peptostreptococcus. Most of these are anaerobes. We're not really worried about anaerobes in a stool. We're actually not worried about them at all in a stool, okay? What we're really worried about is that small group of organisms, most of which are non-lactose fermenters. So we need to be able to differentiate the non-lactose fermenting troublemakers from the normal flora, okay? So that's why we use all those differential plates. We can use in some hospitals, not every hospital, may use enrichment broths, which actually enrich and allow things like salmonella and Shigella to grow up. But for the most part, you're required to use differential plates so that you can recognize those non-lactose fermenters for the characteristics that they have, and say, okay, I'm going to make sure that this isn't salmonella, Shigella, can't be, whatever. Okay. Now some of the other easier sites. And you might think, oh, how could CSF be an easier site? Well, it's not easier on our patient, but for us in the lab, it's pretty easy. Okay, a cerebral spinal fluid is the fluid that surrounds, that bathes the brain and the spinal cord, and it's produced by the meninges. And so when you have meningitis, which is an infection or an inflammation of the meninges, that will release some of the bacteria into the CSF. We collect that CSF and culture it to see what's growing there. Our worst case scenario is if it's something like Neisseria meningitidis, which causes a very, very communicable form of meningitis. Okay, so that's something that we worry about particularly. However, it's not all that common. There are more common causes of meningitis. The most common cause of meningitis is viral, thank goodness, okay? So that means our bacterial cultures won't grow at all. Send it out for viral culture, okay? In this particular case, with the CSF, what is it normally supposed, what's its normal stuff? Nothing. It's not supposed to grow anything, okay? The CSF, its main purpose is to cushion the brain, and it'll circulate the nutrients and remove wastes. But it doesn't have any flora that helps it do that, okay? It's supposed to be sterile. The reason it's particularly sterile, like in other words, it has extra protections over other tissues like blood and the other organ tissues, is because of the blood-brain barrier. This is a vital defense for our nervous system, which is incredibly important to the survival of humans, okay? The endothelial cells that surround the blood vessels that enter the brain and the spinal cord, you can see in this little cartoon over here. In a normal capillary, the endothelial cells, right, these, the blue part here, you have a couple of them, and they have small pores, right, or points of entry or leaving between them. Whereas in the blood-brain barrier, not only do you have the endothelial level, and the junctions between them are very tight. You see there's not that small, the junctions are not easy and loose. In addition, you have these astrocytes which surround, these are the gray, which surround the endothelial cells, increasing the control. So you have like double layers of protection. And you also have these parasites. These are all cells that are able to prevent organisms, proteins, anything really, from passing through a blood-brain barrier. We don't want that to happen, okay? So nothing is allowed through, okay? Now, if you do end up with an infection, it can be a couple of ways. There can be a failure in the blood-brain barrier, in particular as your immune system is affected, okay? You can end up with meningitis, which is an infection of the meninges, or encephalitis, which is an acute inflammation of the brain itself, okay? Both of these are situations that can result in death. Both of these you would detect by collecting a CSF via spinal tap, which the doctor obviously does. And hopefully you'll get a nice clear fluid, no blood, in the spinal fluid so that you can assess and see if there's anything growing in there. You can see here, this is just a little cartoon that demonstrates how the bacteria is able to penetrate through the blood-brain barrier. It's not common. Luckily, this is not a common infection anymore, but it is possible. And it's something that we need to be aware of, in particular for our immunocompromised patients, because those are the people who are most at risk. Now you can see even though there's nothing normal to grow in the CSF. However, there are some ways for us to tell if bacterial meningitis is what we have. Again, I said before, the most common form of meningitis is aseptic meningitis, so-called just because no bacteria grows. But actually, aseptic meningitis is caused by a virus. It's a viral meningitis. And we have clues as to whether we have a bacterial virus before we even start the culture from the cell counts and the chemistry tests that they do on the CSF before it comes to, before micro really can grow anything. Okay, we should get the tube first if there's only one tube because it has to be sterile. But the results will be finished in hematology and chemistry first. And in meningitis, you would have obviously highly increased white blood cells in the CSF. That's a pretty good sign that you have inflammation. But if it's aseptic, then those white cells usually would be lymphocytes, as opposed to bacterial, where the white cells are actually polymorphonuclear cells. They're usually they're usually neutrophils. Okay, so if you have an increase in polymorphonuclear cells, or SEGs, in the CSF, that's starting to lean us more towards a bacterial problem. Okay. Then if we look at the chemistry results, two tests that are done almost universally on a CSF are glucose and protein. If you have a viral infection, yeah, you'll have some increased protein, but your glucose won't be changed. It'll stay pretty much the same, as opposed to in a viral, it might actually be increased because of the increased permeability at the blood-brain barrier. But if it's a bacterial infection, the glucose actually drop, and it's one of the few things that will cause a drop in glucose in the CSF, and that's because the bacteria are eating it, okay? So you have neutrophils up there trying to engulf and phagocytize all the bacteria. You have an increase in protein because of the white cells, the bacteria itself, and antibodies that are being produced, and you have a decrease in glucose. These are the signs from chemistry and hematology that are a red flag to micro, to look for something that might grow. So what are the most common causes of meningitis that we have to look for? And again, some are more common than others as vaccinations become more and more universal. Okay, but the key things that we have to look for: Neisseria meningitidis, Haemophilus influenzae, Type B is the most common one. Streptococcus pneumoniae and Cryptococcus neoformans. Okay. Neisseria meningitidis is a gram-negative diplococci, and you can see it here in this picture inside the segment, the cell. So here's the outside of the cell here. Okay, you can see the nucleus, the segmented parts of the nucleus here. And look, inside, the arrow is pointing to the gram-negative diplococci that are in there. Haemophilus influenza B is it's a very small gram-negative rod, and frequently it doesn't show up on gram stains, so it's actually very light staining. So I don't have a picture of it, but that's a very common cause. So what do we know we need? If it's a Haemophilus, which is fastidious, we must include a chocolate. Then we have the strep pneumo. Strep pneumo is a gram-positive cocci. It occurs usually in pairs, and you can see it here. It's usually lancet-shaped. Look at all the white blood cells. They're all segs, okay? Lots of gram-positive cocci in pairs. And then we have Cryptococcus neoformans. Cryptococcus neoformans is completely different from the other two because it's a yeast. And Cryptococcus does cause meningitis pretty easily, in particular in HIV patients. So this is a yeast that we need to be on the lookout for. When a CSF is collected, again, it's collected by spinal tap. So a needle is inserted into the vertebrae, and a certain amount of CSF is drawn out. Ideally, you can see here, look at this levy. This is a perfect collection. You have a couple of cc's, 2 or 3 cc's in each tube. There are 4 tubes that the physician will collect, and they should be collected in order. These, you can see the yellow numbers on them because for whatever reason, when they manufacture these, there are numbers. These come in the kit, in the venipuncture, in the spinal tap kit, but the numbers are the same color as the plastic. They're very hard to see. Okay. The first tube is the chemistry tube, and that's because chemistry doesn't really care if there's a little bit of contamination by epithelial cells and blood. So we can spin that out, and chemistry can work its magic. The second one will be microbiology because we want the best culture closest to the likely source. Tube 3 would be for hematology, and that's because it's the farthest away. Usually getting 4 tubes is ideal. We rarely get 4 tubes. But if we do get 4 tubes, we like to use the third one because it's really that initial tap, that initial needle stick will have some epithelial cells and potentially some blood cells, and we don't want that for hematology. And then the fourth one, if you get one, is for special testing, cultures, molecular testing, anything like that. Viral cultures, I meant. So this is ideal. 4 separate tubes, each with a few cc's in them. That sounds great. Unfortunately, that's not what we usually get. We usually get 1 tube that we have to split between all of our sections, and micro will always get it first in that case. And a lot of times there's only a half a cc or a cc in there. The more liquid you pull out of a person's spinal cord, the more pressure it puts on the brain because you're actually sucking out fluid, right? You're reducing the volume, increases the pressure. Remember your combined gas laws? So it is a painful process for the patient. It's also a difficult process for the physician, so it's not necessarily something that's always ideally obtained. So that's important to remember. In addition, sometimes the doctor will write himself on the tube where he wants it to go. In that case, you follow the doctor's orders. Now, again, is it viral or is it bacterial? You can see here most aseptic meningitis. You can see we list the viruses that we found out cause it, but look at how many were unidentified. 34% of meningitis, 64% of encephalitis, completely unidentified. Okay, so we're still kind of shooting in the dark sometimes, okay? But it is an inflammatory process. The patient will be pretty critical. And there's a lot of things, viral causes, parasitic causes, spirochetes are another cause. These are not going to show up on a routine culture, so the physician has to take that into account. If you look at this, at the chemistry and the hematology results, aseptic meningitis will produce lab results that demonstrate increased lymphocytes, particularly increased protein, because of course there's definitely inflammation there. But the glucose stays normal. It may actually even rise a little bit, but it won't fall is the key thing. So that's important. The good thing to note is that most cases of aseptic meningitis are self-limiting. I mean, it's going to be a bad few days, but they're self-limiting. So most people can survive them. The bacterial ones are the ones that we're more afraid of. So we always are a little bit more conscious of that. Also, just a word about the parasitic causes. They're not on this chart here. The parasitic causes, in particular, note Naegleria fowleri. They make the news now because people get them from the environment when they're swimming in natural bodies of water, and it can enter through your eye and stuff. That one's a tough one because it's very difficult to treat in patient. It's usually fatal, usually. And for some of these, we have molecular techniques that work very well to identify the viral cause, spirochete cause, spirochete like Lyme's or syphilis. We can do nucleic acid testing for certain markers or gene segments. We could do Western blots. We can do PCR, ELISA for antibodies against them. There's lots of different tests that are available for the most common causes of these diseases. Now we're going to go to wound cultures, which are, to me, the most interesting because they're the most varied. And they're the most varied because you can get a wound anywhere. You get a wound on your foot, get a wound on your back, on your eye, and you get a wound anywhere. The wound itself acts the same, but because it's in the midst of different normal flora, your reading of it has to change with the source, okay? So if you get something, a swab, that just says wound, it's not enough information. You should be calling the physician and say, could you tell me where that wound is so that I know how to read this? Because you do want to know that, okay? And there are a ton of causes for wounds. You know, little kids get wounds all the time. They usually don't break the skin. It's when they break the skin that it can become infected, okay? So occupational hazards, recreational hazards, just life. You know, you get wounds, you fall down, you cut yourself. It happens. There is a population, however, where the wound can start from the inside out, and that's the diabetic population. So we do have to be very careful to know if the patient has those risk factors, okay? Diabetes happens to be one of them. The skin becomes very brittle and dry, and normal flora can actually end up causing an opportunistic infection. By the way, if the wound was caused by an animal bite, that's something we want to know what animal, because then we actually have to refer, all right, what's normal flora? You know, in a hospital that I used to work with, believe it or not, we used to get a lot of duck bites, which it's not really a bite, right? Because ducks don't have teeth, but the moms would take the kitties to the pond to feed the geese. Geese are nasty, and they would bite the kids, and the kids would get an infected wound. But we would want to know that so that we know, oh, there might be duck flora, which is weird, but it might be in there. Okay, so we want to know that. And then if it's something crazy like, I don't know, iguana bite, we might have to look up, well, what would an iguana potentially have, you know? And the vets all know that, so they can tell us what the iguana has, normal flora, okay? Now the best, this picture here is for your purposes to know the difference between the type of specimens that you're going to get. Most common, you're going to get a swab. Okay, that's probably the most common. It's the worst specimen because look at what they're doing. They're just poking at it. Okay, they're just poking at it. Now they probably cleaned this, but if it has this crust on top here, This is scabbed over. We really want to get below that, okay? Which is what has happened in this particular picture, the curettage. I'm sorry, it's not curettage, it's curettage. I can't say the word, but it's a tissue biopsy. Basically, you biopsy the wound. It's very painful for the patient. Pull off that scab and give me a chunk of this nasty stuff. Okay, that's what I want. And that's obviously one. Another one that's really good is an aspiration. Again, painful for the patient. But some of these wounds, you can feel the fluid below the wound. That's probably what they want. So they inject a needle in and they draw out the nastiness, the pus and everything that's just below the surface. And they get that to us. That's what we want. We don't want the swab. If you get either the tissue biopsy or the aspiration, put it in a culture, in a sterile culture cup and send it to the lab. That's what we really want. And we're going to treat it different too. You send us a swab, it's probably going to be treated like a superficial wound. A superficial wound we don't expect too much from, okay? We're just going to look for some of the usual suspects that occur at a superficial level, okay? So those would include things like Staph aureus or Strep pyogenes, which is your group A beta strep. Unless you give me a really good specimen, like either one of these two, I'm not going to look for anaerobes, even though Clostridium, which is an anaerobe, is a very common source of wound infections. But you got to give me a better specimen for me to grow up a Clostridium. I'm not going to try and grow anaerobes on something from a swab because they're not going to survive the swab. Okay? So being able to educate the physician as to, he's saying, I keep sending swabs and nothing's growing. Well, I don't want a swab. Can you send us a tissue? Can you give me a chunk of that wound? Anesthetize the area for your patient and give me a chunk of that wound. So then I can give you better results. The key thing is that we don't really look for anaerobes in many of the specimens that we receive. Like, anaerobes can grow in many places in the body. And because they're normal flora in many places you wouldn't think. You have anaerobes in your nose and your throat. Obviously, you have them in your GI tract, the rectum and the stool. You have them in your cervix. You have them even in your sputum. Your sputum will pick it up as it comes through the throat. So we don't look for anaerobes in those specimens because they're not a common cause. If you have an abscess at any one of those sites, you have to tell us that because then an abscess could harbor anaerobes and we need to look for them at that site. One of the things that we wish that doctors did to every wound is wound debridement. When you have a wound at the surface, and you know this from your own experience, you have a scab. And if as a kid, kids pick off their scabs, right? So the scab has a lot of dead tissue, but it also has a lot of biofilm. The biofilm actually, it's not a bad idea when the wound is healing because it keeps out those nasty pathogens usually. And it will dissipate as the wound heals. So that's a good thing, okay? The problem is because there's an immune response right there, it'll keep the biofilm at bay. When your immune system fails, the biofilm is never controlled. And so we need to remove that using wound debridement in order to do that. It's a painful process where you pull off the dead skin and the biofilm, and you clean it before you collect the specimen, okay? Now, it's actually a procedure that doctors bill for, and it requires a doctor to do it. So sometimes the doctor calls in and tells the nurse, just collect a wound. I want to know what's growing in there. In that case, at the very least, clean the site with sterile saline at least, because you can knock off biofilm pretty easily. It doesn't have that attachment that an infecting organism does with your tissues. You can knock it off pretty easily and then collect the specimen, okay? And that way, what we're hoping is that you're only collecting the infecting organisms. That's really the only ones that we want. How can we verify that? Every wound must have a gram stain. And the gram stain will tell you what kind of cells are there and what kind of bacteria are there. So here's some examples of some wound gram stains, okay? Here's a superficial wound, the first one here. And the reason I know, look at the epithelial cell. That's your epithelial cells are described as looking like eggs, okay? With center small, center yolk, large cytoplasm. That's how you know it's an epithelial cell. Just remember that epithelial cells are flat, and so they fold over, and you can kind of see some of that fold here. They do fold over, though. So they might not always be as giant as this one, okay? And that's okay. You can also see in this one that there's lots of white blood cells, okay? Look at all these segs that are here, okay? Lots and lots of segs. So that's like, oh, wow, lots of white blood cells. Must be an infection, okay? So the other thing you have to look for are the bacteria, any bacteria that you might find. Now, if I'm putting all these dots on here, maybe I'm seeing all these little gram-negative rods or something like that. I would report them. If I see nothing, I report that as well. Because wounds, frequently abscesses especially, harbor anaerobes particularly, we can see here anaerobes. This is probably from an abscess, a peritoneal abscess or something like that. We can see, now you don't know from the gram stain that they're anaerobes. I'm just telling you because this is what anaerobes look like. You see these ones that have these spores. They look like little lollipops or tennis rackets. Those are anaerobes, and they'll grow anaerobically. No epithelial cells in this one. Lots of segs, okay? Now, I know in advance what to expect in the lab. However, what am I going to tell the doctor? For the doctor, for this one, I'm going to tell him, well, there's lots of polymorphonuclear cells. Remember, this is not a Gimza, so you can't call these neutrophils. It's not stained right. So we just call them polymorphonuclear cells. So there's lots of polymorphonuclear cells or white cells. And there's mixed gram-positive bacilli and gram-negative bacilli. So you're not going to try and identify them, but you could say something like many mixed gram-positive and gram-negative bacilli. You might even say spore-forming. That would be fine as well. Depends on the procedures at your lab. In this case here, where am I? Here, this is kind of a mess, but we see this kind of a lot because sometimes the wounds get old. This is a little bit under-decolorized, and I can tell that because the center of the cell is still very dark. Of this glob here, it's very dark purple, and you have a lot of purple going on in the whole thing. But there are lots of bacteria, different bacteria in this slide. And so when you have nothing predominating, and you have gram-positive cocci in clusters and gram-negative rods and gram-positive cocci in pairs, you have all different things, then it is perfectly legitimate. Instead of delineating each one, you just tell the doctor that it's a mixed bacterial smear, which basically means to him, you'll have to wait for the culture results. I can't make a determination at this time. That's different from this one down here. This is on extra high magnification, but you can see this is a neutrophil that has looks like gram-positive cocci inside of it. Now they're in pairs in here. I don't know what the rest of the slide shows, but these are inside the white cell. The fact that the cell is inside the white cell means that the neutrophil sees this as a foreigner. The neutrophil is attacking this. That means we know the target of our infection. So I can tell this physician, I see white cells with intracellular gram-positive cocci. So what does that mean? He knows now that the infection is caused by a gram-positive cocci, and if it's caused by a gram-positive cocci, I can start using my gram-positive antibiotics. Now we move on from wounds to genital cultures, another site that has lots of normal flora. Now, genital culture can mean a lot of things. There's external cultures that you get from genital sites, internal cultures that you get from genital sites. We still expect, even though they're not the same at all, like if you have a lesion, outside on the external structures, you're more likely to have skin flora contamination on a male, genital contamination on a female. You do need to know if it's coming from a male or female, obviously. But we would like the most information that we can. Unfortunately, frequently we just get, John Doe's genital culture. We don't really know much else. So then we just expect normal genital flora. The genital flora for a male and a female are different, okay? They're not the same, so you shouldn't expect the same. The sites are completely different. Makes total sense, okay? What are the sites that you get? Well, in males, urethral prostatitis and urethritis, a urethral specimen, the inside of the penis. The patient might have prostatitis or urethritis. You would get a swab if the physician suspects urethritis. Prostatitis is a little bit different, though, because the prostate obviously is pretty far in. But actually, you could get a semen specimen to look for prostatitis because the semen passes through the prostate. So it will pick up any bacteria that were there. Most of the time, you're just going to get a little bit of skin flora. That's normal. And a semen, obviously semen's produced sterilely, but it's not collected that way. But sometimes, every once in a while, a urologist will send us a semen for culture, and we'll get it, filled with enterococcus or something like that. And instead of thinking that the man has prostate cancer, put him on a course of antibiotics and he's fine in 2 weeks. Okay, so pretty simple solution and much better than cancer, obviously. But what it does is it allows the urologist to avoid the biopsy. Okay. For the female, however, it's different. Obviously, there's actually a lot more sites to worry about because a lot more sites can become infected in the female, and that's because of the close association of all the genital structures with the urinary tract and even with the colon, with the rectum. Okay, it can, especially as the patient gets older, they use diapers in nursing homes, things like that. So unfortunately, contamination from one site to the other is very common. So in the female, there's lots of different sites as well. We would prefer, instead of getting a genital culture, we would prefer if they told us if it's the cervix, uterus, vaginal, ovaries, fallopian tubes. What is it? Is it the vulva? We would prefer to know the specific area. Sometimes I feel like the doctors and nurses totally forget their A and P, and they just say, oh, it's that area. Well, can I have a structure? That would be helpful. Some of the most common infections that you see in the female are bacterial vaginosis, which is usually an infection of the vulva, the external structures. The other things you can have is cervicitis, infection of the cervix, endometriosis, which is an infection of the endometrium. The endometrium is the inside portion, so the portion of the cervix that's inside the uterus. Okay, that should be sterile. It should never be, the cervix technically should be sterile as well, but we do get some transient bacteria there. And then we also have pelvic inflammatory disease, which is an infection of any of the sterile sites, technically, the uterus, the fallopian tubes, even up through the ovaries. Unfortunately, pelvic inflammatory disease can spread through the entire pelvis. It can spread anywhere once it moves in. So it's actually one of those diseases very difficult to identify, but it's important because it can cause a lot of problems for the woman. In both cases, genital cultures, male or female, obviously we have to be able to screen for any STDs that we can grow. Those STDs, the most common one would be, the most common STD is chlamydia. We don't culture chlamydia. We do that by nucleic acid testing. So we can scratch that off our list. We don't have to look for that. The next one after that is Neisseria gonorrhea, and so we do have to be able to culture that. Even though we have nucleic acid testing for that, we do have to culture for that. What the nucleic acid testing has allowed is that the physician can order just the nucleic acid testing, doesn't have to order the culture. However, if he does order a culture, we have to be able to rule out GC, which is Neisseria gonorrhea. So most of these sites, you're going to have a swab collection. Very rarely you'll get a tissue biopsy on any of these sites. The swab is painful enough. So you'll get a swab collection. You have to make sure to include a chocolate and a chocolate plate and also a plate for GC. Most places use the modified Thayer-Martin, but you have to have some. You can use a New York City agar, Gembeck, anything like that, but you have to include one. It's required. Those have to go in CO2, okay? And for that reason, we want that swab as soon as possible. These two pictures here that I included, this is actually from a urinalysis, this one here. But it has something that is seen very commonly in bacterial vaginosis. These are squamous epithelial cells. Hopefully you can see that, but you can tell they look a little different. And that's because they're covered with bacteria. The bacteria are actually kind of attacking the cells, which is the sign of an infection, okay? And what happens in the UA, it's hard to see here, but these will actually change the refractiveness of the cell, and it causes the cell to sort of glitter under the light microscope, and so they're called glitter cells. And these glitter cells, also referred to as clue cells, it's a clue that the patient might have bacterial vaginosis. But we don't see them in micro. They see them in the UA, okay? However, if you do see them, that's a pretty good sign, and it's something that the doctor helps the doctor to diagnose this. This picture down here is from a wound it's a genital wound, unfortunately. It's from a male, and you can see there's lots of neutrophils, right? There's lots of white blood cells, and they have intracellular gram-negative diplococci. Those diplococci are, when you see them inside the white cells, are most likely GC. They're most likely Neisseria gonorrhea. In fact, the rules allow that this only applies to male specimens, and that's because there's so much normal flora in a female that you can't do this little trick with females. But in a male, if you see white cells from a specimen from a genital site, you see white cells that have intracellular gram-negative diplococci, you're allowed to diagnose it as Neisseria gonorrhea and start treatment right away. Again, this does not apply to women. This only applies to a male specimen. Now, it's allowed, but I mean, most doctors will wait for the nucleic acid testing. Just wait for the testing, okay? Most of them just do the nucleic acid testing at this point anyway, because the swab is smaller. So here's the specimens that you're going to see, okay? For the most part, you're going to see culturettes for the micro lab, okay? Now, interestingly, while we do require gram stains for sputums and we require them for wounds, genitals, it's up to you if you want to do a gram stain. They're not all that helpful. And with the advent of doctors trying to diagnose off of a gram stain, some hospitals decide, no, you know what, I'm not going to do a genital. So that's up to the hospital. But best practice in micro is always to do a gram stain. So the double culturette allows you to use one culturette for the gram stain and one culturette for the plates. Now, there's a couple other things that you'll receive in the lab as well. This is a specialized chlamydia transport media. Chlamydia can be cultured, and there's a special swab. You have the male and the female swab. Okay, you got to send them the right swab. But that swab has to go in this special media that'll keep down normal flora, and it'll allow the chlamydia to survive. Chlamydia is an intracellular pathogen, so it's not something that we can grow easily in micro. We need something to support it. Probably the most common one that you'll receive from the genital sites is, if Sloan's a little bit different, but most hospitals receive, is for the nucleic acid testing. The nucleic acid testing for STDs in a genital site will test, and you can see you have male swab, female swab. We'll test for both chlamydia and GC at the same time, depending on which platform you choose. But being able to do that takes out those number 1 and number 2 causes of STDs in the United States today. And so you have a result in 24 hours. And because we don't do sensitivities on GC anyway, it's pretty sensitive still at this point. We don't need the sensitivity, so we just need to identify the patient as either having it or not having it. And remember, at genital sites, we don't look for anaerobes, okay? No anaerobes, please. the doctor would have to explain to us why he wanted us to look for anaerobes. If it's an abscess at that site, we will, but otherwise, no. The only place we would look for anaerobes from a genital site is when you get into the inner sites, so the endometrium, the uterus, those sterile sites where an anaerobe potentially could have been deposited from an internal site. You might run an anaerobe. Otherwise, most genitals don't come from there. We're not doing it. Now remember that the plates, which you're learning, those are your first tool. So you have to know exactly how to use each plate. What am I looking for when I look at this particular plate? The plates that we mostly use, you can cut the blood, the McConkey, EMB, you can look at all these plates. How are the bugs going to grow on each of these plates? Okay, so for the blood, mostly what we're looking for here is obviously growth, but then also hemolysis. That's what we're looking for, okay? This includes alpha hemolysis, red on red, but alpha, beta, and gamma hemolysis, okay? On our gram-negative plates, McConkey, EMB, HE, XLD, all of those, what we're looking for is lactose fermentation, okay? And they each have a different color pattern, but you still should know what the actual colors are for each of them, and you're learning them in lab and in class as well. For the CNA and the PEA, we're looking at gram-positives. Mueller-Hinton is a specialized media that we use for sensitivities. So when you're looking for sensitivity patterns, you get out a Mueller-Hinton. Thio and TSB, those are our broths that we grow up for either morphology or for just to catch everything. Your AJ, KV, and CAMPy, those are your anaerobic plates. So the organisms that are anaerobes will grow there. Yersinia Yersinia will grow specifically on the Yersinia plate, and it grows characteristically with a target morphology. MTM, Thayer, Martin, Gembeck, Neuric City, Agar, these are all for GC. They're all specifically for GC. And LJ, or Lowenstein-Jensen agar, is specifically for tuberculosis, mycobacterium, those organisms. So if I gave you an organism, you should know which plates it would grow on. So for example, Staph aureus, which is a gram-positive cocci, where will it grow? Well, it'll be able to grow on the blood. It'll grow on the other gram-positives, the CNA and the PEA. Pretty much everything can grow on Mueller-Hinton because it's for sensitivities. Pretty much everything can grow on Thio and TSB. And Staph aureus can grow on the AJ, which is for gram-positive anaerobes. The KV is for gram-negatives, and the CAMPy is for Campylobacter. If I gave you an E. coli, where would that grow? It would grow on the blood because most things grow in the blood. It will grow on all the gram-negative plates, McConkey, EMB, HE. It would not grow on the CNA. Okay, and you go on down the list. And those are the things that you're going to have to learn what grows where. As we talked about, the hospital is going to basically develop something that looks like this. It's going to give you the plates that it expects, that the hospital procedure expects you to use based on the source. Okay, so a urine culture, this is a pretend chart, of course, but a urine culture, if you plate that to a blood and a MAC, you're pretty good. A throat, you might plate to a blood and something called a strep screen agar. Sputum, maybe a blood and a MAC, but you also have to include that chocolate because Haemophilus might grow there. Here's the wound, and look what happens in the wound. I start adding my anaerobic plates, okay, and so on down the line. The stool, I have lots of enteric plates because I got to rule out those pathogens. The genital, I make sure I have my GC plate in there. And the CSF, I include everything that'll grow, I have to have my chocolate there, but I might throw in an AJ just in case an anaerobe is there. Your hospital will have their own chart. This is just an example of the things that you might see.